interest of time. As this loads up. Great. So in part one, we spoke about the why, how do we integrate 3D genomics and why? So you've potentially heard from my colleague, Corey. If you haven't, that's okay. I'm going to do a very brief overview of why we might want to integrate data. But for the purposes of this presentation, I'll focus more heavily on the practical aspects of how can we take data from multiple sources and start integrating it into our own workflow. So we're going to talk about integrating multiple genomic data sets. How do we visualize them? Visualization is actually probably one of the most important parts of 3D genomics. It's a very visual data type. And I'll go through a couple examples on how to go through an analysis and draw some insights, taking information from multiple types of data. So this is, my overview of the why, and I think, most of you who are on this call probably appreciate that, whether you're studying disease or something else, in order to get the best picture of the cause of the disease or the mechanism of the disease, we have all these different tools at our disposal, and it's really by looking at the relationship between all of them where we can really develop the most holistic view of what's going on. So on the left, we have, the DNA techniques, talking about the genome. We're really looking at the core order of base pairs. We're looking at the DNA itself, and, we think about this in terms of mutations and variants and what's actually happening to the genetic information. Where we sit in the 3D genome is really how that information is packed and regulated in three-dimensional space. And the epigenome are these auxiliary marks that also play a role in regulating, the access to the DNA, what DNA is available for transcription, and whatnot. So that's the DNA. And as we move towards, the RNA and protein, really now we're looking at things that are the effects of the genome, right? So with RNA, we're obviously talking about the expression of RNA. Protein, more recently now we're seeing genomics extend to the protein level, right? So not only what RNAs are transcribed, but the proteins that are resulting from them, and even further, we're seeing a lot of interesting, analysis assays which are actually looking at small molecules. So not only are the proteins being expressed, but what are the proteins generating in the metabolome and the small molecules? So really, the central dogma of biology is really extending all the way to the metabolites. And the why, really, to sum up part one, the why is, in order to gain a holistic view, we can't really look at these in isolation. It's really the relationship between all these various tools that are at our disposal in order to get the most information about our genotype phenotype that we're interested in. Just to step back and remind, how is it we look at 3D genomics? A lot of this might already be well known, but I'll briefly go through just a review of what is 3D genomics, like what does that word actually mean? And really what we're talking about is proximity ligation at the core of all the different flavors of 3D genomics assays that you'll see. What that means is, we have our ball of DNA. We think of it like a, ball of spaghetti. And the first step usually is to cross-link that DNA, maintain its 3D conformation in space. So things that are in proximity with each other stay in proximity with each other. We fragment that using various enzymes. This is where a lot of the different techniques will differ. Some use DNase. We use both DNase, MNase, and various different restriction enzymes as what you've seen historically for this type of analysis. But we fragment the DNA and then we ligate them back together. We ligate them so that when we clean the DNA, we remove the nucleosomes. We remove the formaldehyde that was used to cross-link. What's left over are these chimeric molecules. So the chimeric molecules, what they indicate are, in the blue here, came from one region of the genome, and in the orange is a piece that was in close proximity at some point and interacting with the blue region, right? So we have these artificial chimeric molecules that represent reads that come from two different parts of the genome. So once we're at this stage, we're at the DNA stage, we have these chimeric, ligated molecules. A lot of what you've seen in the analysis is still holds. We still have to do an alignment. The main difference being because we've artificially created these fragments, if we're doing paired end sequencing, now the paired information, it's not what we generally are used to when we're looking at genomics. We have to sort of ignore the paired end heuristics that comes with doing a paired end sequencing because these two pieces of DNA come from separate regions of the genome. So there's only a minor difference that we do to align this data, but where it differs, from standard genomics is now we have to consider which of these chimeric reads represent valid ligation pairs. So we have a new format that really captures, the chimeric nature of the molecule. It's called the pairs format. Once we have the pairs format, this is where we generate our interaction matrix. And the interaction matrix, in essence, is really just a quantification of how many times two pieces of the genome are interacting with each other. And we do this at various bins. So we bin the genome, and then we quantify the number of reads that describe the frequency that two pieces in distal space are interacting with each other. Right. So the interaction matrix is really the core raw data, if you like to think of it that way. But we can represent this as a matrix. We can represent this as arcs, and I'll talk about the various data types as we go on. So what that means as we're moving from more of a one-dimensional, one base pair or one genomic space, is we need to introduce a secondary dimension to our data type. And that secondary dimension it complicates how we do the data analysis downstream. But that's what I'd like to walk you through today. So, when we're thinking about standard genomics, we got, RNA-seq, whole genome, ATAC, CHIP. We're really thinking about what's the coverage at a single position, and, what is the content of the DNA or RNA at that single position. So we're really only interested for standard genomics in a single position, whereas for 2D genomics, we've introduced now this additional relationship, which is not only single dimension, but double dimension. What is the relationship between two positions in the genome? This allows us to do, various things like, associate SNPs with promoters, figure out, what RNA signals are correlated with, promoters, for example. That extra layer of information is really where we can derive a lot of our new biological insights. From a data perspective, so I'm going to jump more now into the bioinformatics side of this. From a data perspective, we really have to introduce new data types, right? So you're probably well familiar with the single dimension data types. Don't worry about the exact content of these tables. We will be, I think, posting this presentation. So try not to get bogged down in all the tables, but really I want to illustrate the point that, traditional genomics is done in one dimension. We have various file formats for that. The bed file, probably the most basic of that. Applicable to many different data types. For two-dimensional data, we've now introduced secondary layer, and we have, two-dimensional formats. So the BED-PE is, the big brother of the bed file with the additional second genomic location. Pairs format, high-C and cool formats for the interaction matrix. These are specific for proximity ligation data. The SAM file is a little bit of an exception because it can be, the SAM, the alignment file themselves, and it maintains the information of the pair. So the SAM is kind of both. We can think about it in terms of 1D and 2D space depending on how we're looking at it. But because we have these two different file formats, we have to adapt the way that we do these analyses. In essence, this is what they look like at the data level. What's the bed file format has one position. The paired end version of that bed paired end format is really just two positions. And now the key difference is we have two locus, two loci, and some relationship between them. And in general, this is really the interaction or the number of interactions between your left and your right position. So I'd like to now just dive right into some strategies for how can we analyze this data. How can we start taking data from multiple sources, visualize them together, or make new biological insights from them. So I'd like to sort of break these down into three, I guess, thoughts. The first thing we might do is do a visualization. So visualization is what it sounds like. We have these multiple different data types. We have bed files from ChIP-seq. We have bigwig files for RNA-seq. We have our interaction matrix. really the thing that gets us 80% of the way there is just viewing all of this data together. So visualization is a really big component of how can we, make these insights by just by looking at the data. So I'll go over a couple of examples of how we can do that. Comparison. So this is what I like to call, how do we generate hypotheses from our various types of data? Often what you'll have as a result of your experiments are these, large lists of things. We have, for example, for Hi-C promoters that are upstream of loops. Or we might have, open chromatin, a bed file of open chromatin peaks from our taxi experiment. We might have a region of hypomethylated DNA. And the comparison aspect of this is how can we start integrating these? How can we find things that are shared between our data sets, things that are unique to our data sets, and how do we actually do that comparison? And usually the comparison analysis will generate a short list of what we think are biologically meaningful regions of our genome that we want to do further analysis on. And it'll often feed back into the visualization. So, if you already know what you're looking for, you can just load it right up and start looking. The comparison aspect of this is, really to shortlist and, generate hypotheses from your data, which will ultimately go to visualization later. And the third most, I guess, complex type of integration is really a data-specific integration, meaning it's really designed to do a very specialized and very focused integration between various data sets. For example, if you have structural variants and Hi-C data, you might only be interested in new loops that are created as a result of your structural variants. Or if you have Hi-C data and ATAC, chromatin openness, those two things play very well with each other. The three-dimensional structure is also influencing the accessibility of the chromatin. So there's models developed that try to infer what is the cause or what is the consequence of this integration. So data-specific methods, a disclaimer, I can't talk about all of them, but I'll give a couple of examples of kind of what the approach is there and how to start looking at your data using very specific, highly specialized tools. So not to be anticlimactic, but often, the most simple thing you can do is just load up your data into a viewer. Many are probably familiar with the IGV viewer, integrated genomics viewer. It's in the name. The goal of this type of tool is to take your data, which are represented as tracks, and load them all up and start viewing them. I'll just walk through an example here where we have CTCF ChIP-seq data. We have a list of genomic variants. We have various flavors of histone ChIP-seq data. We have methylation data, so differentially methylated regions. We have some micro-seq. So once we, say we're interested in a particular locus because we've identified it as being, highly structurally altered by some whole genome sequencing or something like that. We load up this region in the genome and we can start actually making inferences just by looking at the data. For example, we see, CTCF, we see these, two SNPs that are, expressed right in the middle of a loop. This might point to some potential, non-coding relationship. We see CTCF sites, see two prominent peaks that flank, these two very strong loops that are created. And within this CTCF site, we see acetylation. So we start making hypotheses. This is potentially an enhancer-driven, enhancer promoter loop that's driving expression of, the genes that we see down here. Going further, we can see a large region of hypomethylation or differential methylation. It correlates with H3K27 acetylation, correlates with active promoter. So this is a very highly active, highly transcriptionally active region. And what do we know? We have this large loop that spans multiple genes, so potentially a locus that's very highly active and highly regulated. So just by throwing, our data in a viewer, we can start kind of piecing together, what's happening. This works very well if you have, small tracks that are, at the end of your pipelines, but what if we want to kind of focus a little bit more in and build a little bit more of a I guess, more detailed plot? One of the limitations of IGV, unfortunately, is it doesn't take in high C data. So we do have to start using other tools in order to start incorporating like the interaction matrix. And one of those one of those tools that we use is called Fancy Plot. This is just an example of how we might build this integrative plot using a plotting tool such as Fancy. And again, this is not the only tool available. There are lots of tools that are available, but some of the workhorses in our group that we've come to appreciate, I'm going to go through those. So let's say I'm interested in the MIC locus. Much like how IGV works with tracks, Fancy Plot, you can specify individual tracks using this P parameter, right? So if we're going to look at the high C matrix, we can specify the .high C file at a given resolution for the locus that we're interested in, and generate this plot. We've noticed that there's two very strong loops here. And we've also layered on top of this the TAD blocks. So we know that these are two topologically associated domains that are quite, prominent in our data set. And we're interested in the RNA-seq. We load up the bigwig file so we can, specify a different type of plot. So for the bigwigs, we express these as lines. We can see somewhere here, there's a very strong transcript that's created. We have H3K4 as active promoter, or active marks indicating active transcription at this locus. We have some H3K27. So you see, we can start layering on this multiple different types of information that we have in order to get, really detailed picture of what's going on at our locus of interest. Right. So in this example, an example that we've used quite frequently, this peak in the RNA is a really strong expression of the MYC gene. There's a very strong enhancer at this region. So this is an enhancer promoter interaction, and it's kind of a a less common data type, but pol2 occupancy shows you that it's not only the MYC gene, but this entire locus here is occupied by pol2, indicating active transcription. So like I said, visualization is worth a lot in our data type, especially when you're starting to view different pieces of information together. And you can derive a lot of biological insights just by throwing it into a viewer. Okay, so that's great if you know what you're looking for. If we're a MYC lab and we're looking at MYC, it's easy to kind of go and look at the MYC gene. But how might we start generating these hypotheses, starting from potentially an overwhelming amount of information that we've accumulated through our studies? So I'm going to walk through this hypothetical experiment here. Consider an experiment where we have a wild type and a knockout condition, and we've performed high C on it. And we also have identified, because we're a methylation lab, we've identified a group of highly differentially methylated regions, specifically hypomethylation as an example. Gene we've knocked out is a methyltransferase, so that's why we're interested in methylation. We've knocked out a methyltransferase, and we have this hypothesis that when we do that, we disrupt CTCF-driven loops. And this disruption leads to a difference in expression of some markers that we'd like to identify. Right? So we don't know what we're going after, but we have these various pieces of information, and we like to ask the question, can we identify regions that are impacted by methylation, and can we rank candidates that might be the most interesting for downstream investigation? So an approach we might take are to identify loops that are unique to, for example, the wild type condition. While the wild type, we're hypothesizing that when we knock out a methyltransferase, we've lost methylation, and therefore, by losing methylation, we're going to disrupt loops that are created, right? So we want to basically identify loops that are specific to the wild type condition. And then we want to take those loops and shortlist them to regions that overlapped with a hypomethylated region, for example. So once we have kind of the short list of wild type specific loops that overlap with regions of hypomethylation, we didn't want to perform like an RNA-seq experiment or even a qPCR experiment of our targets to identify which are the genes that are most affected by this change. All right, so I'm going to walk through, example of how we would do this type of analysis. And, many are probably familiar with bed tools. And, you may be surprised that we use bed tools a lot in our work. A lot of the projects that we get coming through coming through the company is actually, this type of comparison. We just want to take two loop sets and figure out, what are the shared loops, what are the unique loops, and oftentimes this is where we start. There are more sophisticated ways to do this. For example, if you have multiple replicates, you can do more of a statistical analysis to get what are the differential loops. But let's just take, this example where we have one wild type sample, one knockout sample, and we just kind of want to do a comparison. Right, so our loops files would look something like this. We have the wild type, we have the knockout, and we can use a command called pair to pair, which is essentially the intersect bed version or the pair version of intersect bed for bed tools. Right, so I'll go through a couple of examples of like how we can use this command. But for example, let's say we wanted to get the loops that are exactly the same. In other words, the left and the right anchor, the left and the right side are the exact same in your wild type and your knockout. You can use a command like this where we have the wild type, the KO. Type both means we're going to find loops that overlap both. So the output of this particular command would be the shared loops. If we want to find not only loops that both anchors are exactly the same, we want to find also loops that are matched on a single side. We can change a parameter in our command here that's either. So either the left or the right can match, right? So that would return something that looks like this. And back to our hypothetical experiment, what we're looking for are the loops that are unique to the wild type. And to do that, we would use a parameter called neither. So neither the left or the right of your wild type and your knockout overlap anything between the two samples. All right, so that would return the loops that are unique to the wild type, which is great. So now we've done that. We've identified loops that are highly specific and only specific to the wild type group. Now we want to do that. Again, we want to take those loops and we want to overlap that with our hypomethylated regions. And the way, there's various ways to do this. We can, again, look at reads that only align on one side or reads that or loops that align on both sides. So using the type either would return, this loop and this loop because both sides overlap here and one side overlaps here. So we'd get both of those. If we use the type both, only the loops that overlap on both the left and the right side would be returned. So depending on our hypotheses, we might choose a different strategy for how do we overlap our loops in our regions. And in this case, as the command suggests, we're using a pair file against the bed file. So this is a two-dimensional file. We're just overlapping that with a single-dimensional file. Okay, so great. So this is what we've done. We've identified the loops that are unique to the wild type condition. We've identified of those loops, which ones overlap with the region of hypomethylation, because that's what we're studying. We've knocked out a methyltransferase, so we're only interested in the regions that overlap with some effect on the methylation. So we've knocked off point 1 and 2 of our approach. Third step is, okay, we've identified this short list of genes. Now let's try to rank them and see which ones are most affected by the change. Right, so what we'd have to do is, because we're working with a region, we'd have to annotate these with genes, and then we'd have to rank them by some method. I'm only showing one method. There's multiple ways that we could potentially annotate, a list of regions. But because we're working with loops, it's very similar to, I think, what you'd see in the ChIP-seq literature, where you have a peak and there could potentially be multiple genes that are within the peak or upstream of the peak. But we can borrow from that literature and only focus on genes that are potentially upstream or in the promoter region of these loops. So genes most likely to be affected by these loops. And this is an example of how we might do that. ChIP-seeker is borrowed from the ChIP-seq literature. Really what we're doing is we're defining what genes we want to look at. We're using our bed file. So this in red is the bed file that we've identified in the star region here. We're giving that to ChIP-seeker. We're annotating plus or minus 1000 kb at the promoter and returning the list of genes that are likely to be at that promoter. Right, so the last step, we've identified, we've ranked, or we've identified genes likely to be impacted by this loop. We might choose to add RNA-seq or qPCR, depending on how long our list is. And what we'll be able to do is kind of rank these. So if we did RNA-seq, we might now have an FPKM. And we might be able to look at in the wild type and the KO which genes are the most different, right? So we can apply a very simple statistic. Let's identify, the largest difference between the two conditions and start ranking our genes that way. You might not get the exact answer at this point, but you've at least shortlisted your data, right? So what we've done now is we've identified a group of genes that are regulated by loops that we know are disrupted by 3D structural changes induced by our knockout. Right? So it's kind of a mouthful, but you can see how we've incorporated multiple types of data and really said a lot already about the biology. And again, as I alluded to, now that we've identified these regions, then we'll start applying our visualization tools downstream. So this is an example of a group that did basically exactly what I'm speaking about. This is a paper by Flavahan that was spoken about in part one. Very sentinel paper that really outlined a really nice phenomenon where if you have a CTCF loop, this can act as an insulator to upstream or downstream enhancers. But if you methylate that CTCF site, it disrupts this insulation, thereby activating enhancers that are downstream that are normally not acting on the locus. So what is the approach that they take? And essentially what they did is what we spoke about, identify differentially bound CTCF sites. These they've termed lost insulators, right? Because they're hypothesizing that if you've lost the CTCF interaction, then you've kind of lost that insulation. You overlap those sites with methylation, so that's figure C here. They've change in CTCF binding, so we have our difference in our CTCF sites, and in red we have the regions that are methylated, right? So they'd be interested in all these regions that are in red. So this is your Venn diagram, and then they've ranked them somehow. So they have these regions of CTCF lost insulators overlap with methylation data and have been ranked now. So they're not using RNA-seq to rank them. They're using H3K27 acetylation. And so they're looking at, of all those regions, let's quantify the acetylation signal in that domain, in those TADs. What they found was the ones with the highest score, they found a number, HOX1, KIT, FGF, PDGFR-A. So they still don't have the answer, but then they layer on another piece of information. Now they layer on the RNA-seq, and what they see is, well, KIT's not one of them because it's not much change between my condition. It's actually FGF, and FGF3 and FGF4 are quite heavily upregulated in the data set. So you can see how this strategy really takes a lot of information. This can be potentially hundreds of thousands of CPG islands, and shortlisted it to a number of genes that really potentially influence our experiment. All right, so that's an example of a comparative analysis. I'd like to end off, just check how we're doing for time. I just want to end off so we have some time for questions on the data-specific methods. And really, these is probably one of the most exciting aspects of the field right now. Is there's new models that are being generated that are really specific and really designed to make a specific inference based on multiple different data types. So this is just an oversimplification of kind of the approach there. Is you have potentially 20 different data sets. They've developed some sort of model that leads to some sort of inference. So I'm going to go through just two of those very quickly. One of them is called TEPIC. TEPIC, the whole goal of this technique called TEPIC is to identify likely transcription factors that are active in your sample of interest. And ATAC-seq combined with Hi-C is actually a very powerful method to predict which regions are potentially active with transcription factors, right? So if the chromatin's open and there's potentially a loop that's causing it, that's very strong indication, strong evidence that there's potentially a transcription factor there. And then taking into account some of the motifs within that transcription factor in those regions, we can develop very nice prediction of what transcription factors are active. So a lot of these models are coming to light now, and this is just an example of how to run TEPIC. You have a region of open chromatin, you have your loops, and so ATAC-seq as a bed file in green, loops as a bed PE file in red, and we throw it through their model, and at the end of this type of analysis, what you get are for every region in your ATAC-seq, what are the most likely transcription factors that are active. Right, so very, conceptually very simple. There's model at the core of our data type that's really designed to do one specific thing. Another example of that is, combining structural variants with our Hi-C data. Right. So for example, if we've identified a list of structural variants through whether that's whole genome or whole exome or copy number variants analysis, and we have Hi-C data, we can actually identify potential candidates of enhancer hijacking. Right. So Neo-Loop Finder is one of those tools that is available that helps us to do this. Right. So we have a list of SVs. This is SVs in BEDBE format. We can assemble those SVs into what is the predictive kind of complex structure, right? Because now with Hi-C, what we have is not only the structural variants, but how they are linked in space, right? So we can assemble complex SVs. We can then eventually call new loops that are created. So say we have an interchromosomal translocation between two different chromosomes. We can assemble that into kind of a new reference. And then with that new reference, make a prediction about new potential regulatory elements that are created as a result of it. Right, so here's an example of the MIC-PBT locus. We have, MIC and PBT are kind of far away in three-dimensional space. If there's a structural variant, you can see that there's new loops created that are potentially regulating this new locus. This is another one with the NF-kappa beta gene. If you have RNA-seq and acetylation, you can actually go a step further and say, not only have we created this new regulatory loop, but hey, there's expression of this gene and there's an enhancer upstream. So we can actually explicitly call these, events of enhancer hijacking. But again, another example of a highly specific kind of tool that really is designed to take in two types of data, SV and Hi-C, and do something very specific, which is called new loops. So these things are, constantly emerging. I think it's an exciting time, and you'll see a lot more of these emerging in the years to come. Okay, so I'll stop there. Integrating multiple data sites is probably the most powerful way to develop a holistic view of the genome. Visualization, I hope I've kind of made the point that a picture is worth more than 1,000 words. Comparison leads to powerful testable hypotheses that we can use to take multiple inputs, generate a hypothesis, shortlist our data into things that we can actually go forward with and test. And there are lots of emerging methods specific for how do we integrate 3D genomics with other data types. And I think, like I said, it's an exciting time. And hopefully, these are the tools that we've used in the presentation, but hopefully there's some time now for some questions. So thank you very much.
Great. Thank you so much, Jonathan. Wonderful presentation. If anyone has questions, please go ahead and post them in the Q&A box. You can see that at the bottom of your screen. All right, there's one for you. Would you like me to read it, or do you want to?
This one looks like it's for you. Our pleasure.
Oh, yeah, exactly. Thank you. Yes, I will send out the recording in about a day or two just to edit that and send it out via email. So for the email you use for the registration, I'll send the recording out. All right, we've got another one.
I have another question. Do you also analyze RNA's M6A modifications? So I probably need a little bit more context to the question there. Perhaps something that, if you have a specific question on how to do a specific integration of your data, we'd be happy to kind of answer that. But if you do have any more information, what do you mean by analyze? I'd be happy to answer. So I'm not quite sure. We in-house don't analyze RNA's modifications, but I'm sure that the integration with 3D genomics would be quite possible, and we'd love to work through that.
And if you want to add more information to your question, you can go ahead and do that. All right, we'll just give it one more minute. Here we go.
Another question, to get a good understanding of gene regulation, how many histone modifications would you suggest? I mean, definitely, a very project specific question, right? If you're interested in gene regulation and overexpression, then you might only need acetylation, right? Yeah, we showed in the MIC example acetylation plus K4 trimethylation plus pol-2 occupancy. Yeah, that could potentially be overkill depending on what your question is. But a good strategy is to start with one because that will lead to a lot of hypotheses. And then depending on your findings, you may wish to add additional layers on top of that. And I think that's a good strategy because number one, saves a lot of money. But number two, you don't want to kind of go in with 5 different histone marks if only 2 or 3 of them are relevant for your study. So my suggestion would be to start with one, probably the one that you think is the most important for your specific study and go from there. Um, I can add on to that acetylation is probably the most relevant when you're talking about enhancer promoter loops. So, I think acetylation is a really powerful one, but also other transcription factors, ChIP-seq experiments are also good to do as well. Another question, you talked a lot about loops. How many total loops should we get from experiment? And what's a good start to run that analysis? It's a great question. We have kind of spoke about the levels of depth that we generally require for our experiments. If you're doing, whole genome micro-C, we're generally looking for, 800 million reads will give you, thousands of loops. But it is really highly dependent on your sample, and we do have very detailed quality control metrics that you can refer to with um, our lab folk. So there's not a one answer to that. It could be thousands, it could be hundreds, and both of those could be valid. But we do have good ways to gatekeep QC. Another question, in your experience, how much time should one plan to spend on the Hi-C plus ATAC-seq integration analysis? Is it highly time consuming and computationally intensive? That's another great question. And I think a lot of the tools that are emerging, which are model based, where they've developed a machine learning model for the integration of multiple data types, they're actually quite lightweight, right? So the one that I showed, which is ATAC plus Hi-seq, it completes in like less than a minute. It's really fast because that model's already been generated. If you're going to develop your own models, if you're going to be the next publish the next kind of integration model, then of course developing those machine learning models are going to take more time, and that's where the time is. But thankfully these things are starting to emerge and become a bit more available to us, and once the model's developed, it really is a low cost compute. um, yeah, so the example I gave is really low cost. There might be more, but it is, once the model is generated, highly doable. um, Yeah, question about SVs and neo-loop calling. What coverage do you recommend for this analysis, and what size of bin is needed to do so? All right, so for the example with neo-loop finder, they actually have a tool that works upstream of that called EagleC, which calls structural variants from the Hi-C data. If you're going that route, then you probably need enough coverage in your Hi-C data to call accurate SVs. And in our hands, generally that is on the order of 20 to 40x is a good amount. If you're looking for, really small intra-chromosomal things, you have to sequence a little bit more probably. But yeah, and 20 to 40x is more than adequate for these large complex structural SVs. And what bin size do you need? So if you are in that 20 to 40x range, you're generating bins, that's 10 KB and above for sure. And the Neo Loop Finder does it at 10 KB, 50, and I think 1 megabase. So the lowest you need is 10 KB, which is well within the limits of the data type that we work with. Um,
Yeah, there's a
I think
couple I think that got missed.
I missed some. Okay.
Yeah, if we go back up through, I
So
think.
yeah, one I see is a good reference that uses these integrations. Yeah, so if you actually look at part one of this talk, I believe that will be posted. Corey spoke about a number of um, number of publications that do exactly kind of the stuff. I showed one, which is the Flavahan paper, which is, the methylation CTCF occupancy one. But there are a number of references. I'm sure we're happy to pass along to you. I think, did I miss
Yeah,
some?
and all of those talks are up on our website under webinars, and or videos, and you can find them there.
Right, so moving on, does capture micro C with human pan promoter? Is it a better choice to call more interactions? I think if, so I think, it comes to a question, when do you do whole genome versus when do you do a capture? And again, it really depends on your experiment. If you are capturing something, you're obviously going to get a lot more coverage. So you have the potential to, call loops with higher statistical power. Doesn't mean, like you said, like you've identified, doesn't mean there's going to be more loops because you are capturing just one space. Micro C is almost always going to find more loops because you're looking genome wide. But again, the statistical power within each loop is going to be lower just by coverage. It's very similar to what you would do for a targeted sequencing experiment. You're going to get more coverage, but you're going to find less. I think it depends on your experiment. If you are only interested in a specific region, then capture's a great choice. If you do want to take more of a de novo whole unknown approach where you don't know what you're looking for, you have two conditions, but you don't really know what you're looking for, then micro C is going to be a good solution there. For high chip, would you recommend performing biological replicates for calling loops? Common set. Yeah, so we do have tools that are and they're not us, but there are tools available for doing like differential analysis, and we do run that as well at Kintata. Generally, when it's capture, we recommend replicates because the tools downstream that are doing the differential loop calling depends on having replicates. So there's not necessarily in that case, a merging of loops or anything. It's actually all kind of contained within the statistical tools that are doing the differential analysis. Um, so, for differential, there are tools specific for differential. For the case that I showed in my talk is like where you have one and one, where you can't really do a differential analysis, or where that potentially might be overkill, or you've downloaded an external data set and you don't have the raw data to do that. You might do this very simple bed tools kind of integration, or you might do a differential and then you downloaded, ATAC-seq or H3K27. You have these two lists now that you want to kind of compare. So that's kind of, and you'll be surprised that we do a lot of that. And that's, the paper I showed did exactly that. It's really just comparing lists. So, you in your study, you'll probably use a combination of a tool specific for differential plus kind of integrating various different lists. So for high C and ATAC-seq analysis, is it better to have a high sequencing depth, but less biological replicates, or low depth, but more replicates? Yeah, that's an interesting question. What I would say is you probably want to do for both of your high C and your ATAC, because high C depends on loops, or the analysis, the integration that I spoke about at least depends on the loop calls. And the ATAC-seq really depends on strong peaks. You're going to want to do both experiments at an appropriate depth to get good peaks and to get good loops, because that's the input. You're not inputting the raw data themselves, you're inputting actually the calls from the raw data. So the question of replicates versus sequencing depth really depends on your study. And, it probably an unsatisfying answer, but you probably want to do both studies at the depth required to get the calls that you want, right? So for loops, loops is the highest resolution kind of call that we get from high C data. You will need adequate depth so that you can generate good loop calls. And for ATAC, I haven't done a lot of ATAC recently, so I'm sure the guidelines have changed, but you would require enough depth in your experiments to get the quality peak calls that you need. Moving on, any suggestion for tools that statistically integrate duplicated micro C and omics data? Sorry, I'm not exactly sure what you mean by duplicated micro C, but I think you mean if you have two different micro C data sets, how to sort of integrate those. And, I think the pair tools method that I showed is a great starting point for how to do that, especially when you have microC from two different data sources. You can work with the calls directly. Um, but there are also tools if you have, microC replicates where you can sort of, merge microC data into a kind of a singular interaction matrix, right? So you can do some statistical um, methods on the interaction matrix files themselves, calculate fold change between two matrices, etc. So there are ways to do that. Again, it kind of depends. And I think, a lot of these questions are kind of highlighting on all the different types of things that you can do. There's, it's very hard to kind of hit on all the different integrations, right? There's potentially 20 different types of genomics tools, and it's hard to hit on every type of integration. But the pair tools, list integration is really fundamental to a lot of them, right? At the end of the day, we're still working with bed files, bed PE files, wiggle tracks, and at the end of the day, the tools that are used to integrate and to find relationships between them are going to be similar. Please suggest some tools to do HiChip. Our tool of choice is Chicago. We use Chicago to do, sorry, HiChip. So Chicago is for capture. HiChip, we use a tool called Fit HiChip, which is designed to call loops from HiChip data. There are others that's our tool of choice. It seems to work really well. And the nice thing about Fit HiChip is it has you can follow that with differential loop calling as well. It's got a module that allows for that. But as for upstream processing, a lot of the tools that are used for microC and other types of Hi-C restriction enzyme tools are still going to apply. So, we use a combination of BWA and pair tools for our kind of Hi-C workflow. But, Hi-C Pro and all the different larger suites are still valid as well and give very similar data. So for HiChip specifically, Fit HiChip, it's our recommendation, and everything upstream, BWA pair tools is what we do. Thank you.